Hi, everybody. So we're going to start microbial genetics in this chapter. It's broken into four sections, so it's quite a long chapter. Just get ready. The first thing we'll talk about is the central dogma, which is the transfer of information from DNA to RNA, which is a different kind of molecule, and then translation, which is where the information in RNA gets used to make protein. So the flow of information goes DNA to RNA to protein. The two steps that you see are the things we'll be looking at. So the first step is called transcription, and that is taking the information from DNA, creating a molecule of RNA, and then translation is taking that RNA and making a different kind of molecule altogether, which is a protein that's made of amino acids. So this is some terminology you should know. A genotype, all living things have a DNA as their sort of hereditary material, right? So the genotype is basically what genes do you have, what kind of different genes that are in your total genome. And then your genotype can interact with the environment. So we have environmental conditions A, environmental conditions B. So you can have one set genotype. Given the conditions that this cell or the organism is in, you can actually have different expressions of the genotype. So you can get the phenotype A, or you can have phenotype B. So what's the phenotype? A phenotype is what you physically make from the genome. So think about the genome as a library of all the information there is. And then the phenotype is like, what information are you accessing from that library? What genes are you going to express that make proteins that will make the cell do different functions or look differently? And in turn, the organism may look different and function differently. So the environment can actually influence, right, what genes are being expressed or not expressed. So that's basically that lesson here is the genotype is the genes you have in your DNA. It's the order of the A, T, C's, and G's. And then the phenotype is the expression of the genes. So what genes are being used, how much of the proteins are being made, which proteins are being made, things like that. All right, so genetics is the study of the transfer of information between biological entities, right? So it can be cell to cell, it can be virus to bacteria, it can be plant to plant. And when you talk about an organism's genome, it's basically the sum of all of the genetic material inside the cell. Most of the genome that we're used to, right, exists as chromosomes in the nucleus for eukaryotes, or we know that bacteria, prokaryotes, have an essential area called the nucleoid, right? But it's not membrane bound because prokaryotes, again, do not have membranes inside their cells. There are also other non-chromosomal sites, so DNA that's also found in the mitochondria. We talked about that, how mitochondria were thought to be free-living parasites a long time ago. They do have their own DNA. mtDNA stands for mitochondrial DNA. The chloroplasts that photosynthesize also have their own DNA. And plasmids, we'll talk about plasmids in this chapter. And plasmids are also little circles of DNA, and they end up being sort of bonus DNA genes. Okay, so this is a basic difference between prokaryotic and eukaryotic genomes, right? So eukaryotic genomes, we have many linear strands of DNA, which we call chromosomes. So, we have 23 pairs of chromosomes for humans, right? So we have 23 pairs of linear strands of DNA inside our nucleus. For prokaryotic cells, they generally have one circular chromosome, but there are some exceptions. But really, for this class, we'll just think of it as one circular chromosome. In eukaryotic cells, our DNA is packaged around a particular type of protein called histone proteins. And prokaryotic cells also organize their DNA around a protein, but they're not histones. So they are condensed and secured with other proteins, just not called histones. And of course, we have this in a membrane-bound nucleus, and then this is in the nucleoid. You want to know the structure of DNA for this next upcoming test. So DNA is a double-stranded, double helix, helical shaped molecule. And the next thing here is an anti-parallel arrangement of strands. So this is going to take a little bit of explaining. What that means is, if we look here, so I'll start explaining what a nucleotide is, right? So this unit here, all right, is called a nucleotide. A nucleotide has three parts to it. It has a phosphate group, it has a sugar, all right, and this sugar happens to be deoxyribose. So that's where DNA gets its name. The sugar is deoxyribose. So these two structures, the phosphate group and the deoxyribose sugar, make what's called the backbone. So if you imagine this is like a ladder, right, you would step on our bases here, or A, T, C's, and G's, and then the sides of the ladder would be these phosphate groups and sugar groups. The bases, so back to our nucleotide, the nucleotide is three parts, right? Phosphate group, in DNA's case, deoxyribonucleic acid, deoxyribose, and then we have a base. So these are, the long name is a nitrogenous base. There are 4 different bases in DNA. There is the T, which stands for thymine. There is the A that stands for adenine. There's the C and G, which stand for cytosine and guanine. And notice that they do face each other inside of our DNA molecule, and the little dots here represent the bonding between those bases, and those are hydrogen bonds. So you really want to know that the hydrogen bonds are what's holding them together. Another thing to remember is that these hydrogen bonds are relatively weak, okay, relatively weak. Of course, they're not so weak that DNA breaks apart all the time, but you have the ability to break these bonds when you need to copy DNA or when you need to express DNA. So just something to keep in mind. Now, back to the anti-parallel arrangement, okay, this word anti-parallel. Notice on this strand, right, so one side of DNA is called one strand, and this is the other strand, and that's what we call DNA is a double-stranded molecule. And then when you twist this up into basically a corkscrew shape, right, that's our double helix shape. So the 5' and the 3' refer to the orientation of the sugar. So real quickly, in organic chemistry, when you start to number the carbons in the sugar group, when you draw something like this, each corner represents a carbon. So the way you number it is like this is 1, 2, 3, 4, 5. Oops, sorry. 1, 2, 3, 4, 5. Okay, so this is our 5th carbon. And you see how the 5th carbon is attached to the phosphate group, and there's nothing else up here. So this would be like the end of the DNA strand. So we call this the 5' end because this 5' carbon is not attached to anything else. There's nothing else above it. I mean, it's attached to the phosphate group, but there's nothing else above it. If we look at the 3' end, it's different, right? So again, there's 1, 2, 3. This is the 3rd carbon. It's not attached to anything below it, right? So that 3rd carbon we can add. If we look at the 3rd carbon here, right, you can see that we don't see the OH because it's been used to attach to the nucleotide that's down here. So this is the 3' end that's attached to another nucleotide. So this is the 3' end because it's empty, right? So if this was a complete molecule of DNA, one strand is going to have the 5' end free, meaning that you can build above it, and then the other end will have the 3' end free. Now, anti-parallel means the other side is opposite, where the 3' is on one side and the 5' is, so it's kind of like a flipped or like a 2-lane highway where the cars go in different directions. So 5 to 3 goes this way on one side, and then 5 to 3 goes this way on the other side. That's all it means, anti-parallel arrangement. So this is what our double helix looks like, right? I'm sure you've seen this molecule before. Again, the backbone is the sugar and phosphate. Remember, the sugar in DNA is our deoxyribose. And in RNA, it's different. In RNA, it's just ribose. So the difference is basically this little OH group down here, but um, just so you know. So there's two different sugars, one for DNA, the deoxyribose, and the one in RNA is just ribose. So I'll spell it RIBOSE, ribose. And then we can also see in this picture that the bases, right, these nitrogenous bases are facing inwards. And there's a pattern to how they bind to each other. So A always binds with T, and then the G's and C's are always going to match. So you can think about the two like curved letters, right? They're always going to match each other, C and G. And you can see that's happening here too, right? C and G, C and G, A and T, A and T. So that's how they pair bond or in DNA. Okay, so the basics of DNA is there. For replication, when you want to copy DNA, right? That's what we're talking about. We want to copy the DNA. Why would a cell want to copy all of its DNA, right? And this happens only when the cell is going to divide, right? Because if the cell is going to make daughter cells, right, and divide in a very asexual way, which means that there's no genetic recombination, that the parent cell and the daughter cells are clones. They're exactly the same. They have the same genome. You have to copy all the DNA here, right? So you have two copies, and then one copy goes here and one copy goes here, and then you have two complete cells with the entire genome in each one. So how is DNA copied? All right, so DNA replication. There's a lot of information here. When scientists were starting to figure out how DNA is copied, they had basically three choices. Conservative, meaning that you're going to conserve. So this light blue is going to be the parent strand. So you have two original strands of DNA. And then if you were to copy this, the one idea is that both of the parent strands are kept together, and then these newly made strands are on the other side. And so these two molecules of DNA, one is all conserved. So conserved, meaning that we use the same two parent strands, and the other one is completely new, put together with nucleotides. It turns out that that's not the way that DNA is made. So it's made in this semi-conservative way, which means that we take apart these two parent strands of DNA. Remember, there are hydrogen bonds between them, so it's easy to break open. There's an enzyme that does that. And then when they're split open, this parent strand acts as a template, right? It acts as kind of like instructions on how to make the other strand, which is actually quite simple, right? So if we go back to, if we had a single strand of DNA, remember we have our A's, T's, C's, and G's. So let's just do a little strand of DNA, right? So let's say we have in one strand A, T, A, C, C, G, right? You know what the other strand is. You know that the other strand is going to be a T here, right? So if we were building a new strand of DNA, we're building a molecule of DNA and we had just one strand, we would know the other strand because of that pair bonding rule. If there is a T on one side, you know there has to be an A on the other side. If there's an A here, we know there's another T here. If there's a C, we know that a G is going to go there. A C, we know a G is going to go there. And then if there's a G, there's a C, right? So it's really easy. If you take apart the parent strands, you can read one strand and it acts as a template to create another strand. And so that's what happens. We can see that we have a replication happening here. We conserved one parent strand in the new molecule, right? And the other strand is completely new. So red indicates that it was put together by an enzyme using the building blocks, our A, T, C's, and G's, our nucleotide building blocks. And then the other strand is the same way, okay? So that's all you need to keep in mind is that the way DNA is made with the semi-conservative replication, where a parent strand is going to be separated and it directs the synthesis of the daughter strand. So in this case, right, the red would indicate that's the daughter strand. All right, so we're not going to really talk about dispersive. There's a lot of information in this chapter. We do need to leave some things. So keep in mind that DNA replication is semi-conservative. So we're going to have to look at how it's done. There's a lot of enzymes you're going to want to know for this replication process. So The most important enzyme is going to be DNA polymerase 3. There are other DNA polymerases, there's 1 and 2, but I'm not going to have you guys know them. It's just going to be DNA polymerase 3 for us. And it's responsible for adding those nucleotides to the growing new DNA strand. So in essence, right, this enzyme can detect, if we had an A on one side, if we had like A, C, G, C, something like that, this enzyme can detect the A, and then you would just have to believe, right, that there's all kinds of building blocks floating around. And the enzyme, DNA polymerase, is able to attach the correct nucleotide to the opposite side, right? And if there's a C, this enzyme is able to detect the C and it's going to add the complementary G. If there's a G, it's going to add a C. If there's a C, it's going to add a G. So it has this mechanism that's able to do that, right? So that's how it will add the new nucleotides to the growing new DNA strand. What's really cool about DNA polymerase also is that it can detect if it did put in the wrong nucleotide by mistake. It can sense it. There's going to be a kink in the molecule. It's not going to feel right. It's not going to be a good fit. So it can take it out and then it can put in the correct one. So we call that proofreading ability. So it's really nice that it does that, because if you think about if it did make a mistake and the mistake stayed in the new molecule, then that would be a mutation, right? A mutation is any change in DNA. And a lot of times mutations are bad for the cell. So the fact that it can proofread is great. And it proofreads in both our eukaryotes and our prokaryotic cells. So DNA polymerase can fix the mistakes. And then in this case here, this cartoon, it says, "Hey, Jerry, can you take a second shift today? Sorry, boss, but I can only do the 5 to 3. So this is getting to the next idea, which is that our DNA polymerase enzyme is only allowed, or it's only able to add in this 5' to 3' direction. Meaning, remember that 5' end and the 3' end and how they're chemically a little bit different. DNA polymerase can't go both ways on the strand. It can only travel from 5' to 3'. Okay, so just keep that in mind. So we'll talk about replication of eukaryotic DNA first. So the way that our DNA, because it's a linear strand, right, it's a string, sort of, it's going to be different than bacteria, because bacteria have a circle. So these, I've listed all the terms that you want to be familiar with in the process of replication of eukaryotic DNA. So let's go through them. Here is our strand of DNA. It's a double-stranded, so those little bars in the middle, they're going to represent the bases. And we can see that we have these bubbles opening up, right? The place where bubbles begin to open is called the origin of replication. This is actually a little bit of a sequence that the enzymes, when they come to open up the DNA, they recognize this origin of replication and they start to open up the DNA. Our DNA is really long. It does not start at one end and just go straight through, because if it did that, it would take way too long to make a copy of DNA. So what happens in eukaryotic cells is that there's many, many origins of replication. And so you'll start to copy DNA just all throughout in little bubbles. And so those are called replication bubbles. So those circular open areas is a replication bubble. And the word replication fork is just the corner here, right? So if a fork in a road, like if you hit here, this is our fork. And so the arrows that you see here indicate that you can actually copy DNA in both directions. So as this bubble opens, the enzyme DNA polymerase will attach and then just begin to make that new strand, that daughter strand, which is indicated by this dark red. And it'll start to move in one direction. And then also, so there's not just one DNA polymerase, but there's many of them. So one will attach here, start to go in this direction. One will attach here and start to go in this direction. So you can imagine that sooner or later, you can see that all the bubbles start to get bigger. They're more, DNA is getting made. And pretty soon the bubbles will all just meet and you'll have that new copy of that new DNA. And so you can see down here, right, the parent DNA is our light blue DNA, and then the newly made is red. And we have two molecules, right? So one molecule here, another molecule here. okay, in this semi-conservative manner. Now, there are some more things to talk about, and there's pictures, so I'll go ahead and just show you the pictures and come back to this list. So this is what one replication fork looks like, right? So we're just looking at one bubble that's been opened, and we have this nice enzyme here called helicase. So remember, DNA is a double helix. So helicase is going to break open that helix and break those hydrogen bonds so that the two strands separate. And we have one helix working on one fork. If you imagine there's another helix working on the other side, and they're just opening up that replication bubble. Okay, so here's our DNA polymerase. This is representing the enzyme, and it can just latch on to one of our parent strands and start to basically recognize the parent strand and go ahead and add on the complementary bases. Okay, so because DNA is anti-parallel, and also because DNA polymerase can only go from 5' to 3'. Okay. So look at the 5' end here. The 5' end is what's being attached, right? So it's starting at the 5' end and it's moving towards the 3' end. On the other side though, because DNA is anti-parallel, it doesn't work the same way because this strand is moving in the same direction as the fork is opening. So you can imagine that this guy, he just has to scoot along in one direction, and as he scoots along, this helicase will open up more and he can just like keep going. But if you're on the other strand, here's the 5' end, right? It's moving towards the 3' end. But the helicase is opening this way, right? And this guy is moving this way. So what happens is this will attach to a little area and start to copy the DNA in this direction, right? Add those nucleotides in one direction. But then you have this helicase opening the opposite way. So you're going to have a gap. You're going to have a little bit of area that's like not made, right? It's empty. So what ends up happening is another DNA polymerase will attach here and then start to move in this direction. Okay. So on this strand, it's made in little snippets because of the manner that DNA polymerase can only move in one direction and the two strands are anti-parallel. So what we call this strand that's basically doesn't have any issues, the DNA polymerase moves in the same direction as the fork, we call that the leading strand. And the other side, because it's anti-parallel and we're making this strand in little fragments, this is called the lagging strand. Okay. So the little fragments that are made by this DNA polymerase, they're called Okazaki fragments, okay, because the scientist who discovered that is named Okazaki. It's a fun word to say. And so that's basically it. So let's go back here. I did leave off an enzyme. So helicase is the one that unwound the DNA and broke those hydrogen bonds. We talked about the leading and lagging strands, and we talked about Okazaki fragments. Now let's talk about primase and ligase and then the telomeres. Turns out that when you open DNA, this guy here, this DNA polymerase, cannot attach to a single strand. So it has to have a double strand to attach to. So it seems kind of like fussy, but there's another enzyme that comes in and puts like a tiny little double strand in so that the DNA polymerase can attach to it. Okay, so again, DNA polymerase cannot attach to a single stranded DNA molecule. It has to attach to a double stranded DNA molecule. But since we're opening it up, there's another enzyme called primase that comes in, makes a little bit of a double strand for this guy to latch onto so that it can go and do its job. Okay, so the primer is just that little strip of extra DNA to make a double strand so that our DNA polymerase is able to attach. Same thing happens on this side. You see there's a primer, right? So DNA polymerase, again, is limited because it can only move in one direction and it can only attach to a double-stranded DNA molecule. So a primer is required. and it basically directs the DNA polymerase where to attach. And so that is done by the enzyme primase, right? So there's primase. Primase adds that double little piece of DNA so it can the DNA polymerase can attach. Now, our last enzyme is called ligase. I don't think we see ligase on this picture, so ligase is mentioned here, but I'll just go back to our first picture. So ligase, what it does is because the Okazaki fragments are not going to be complete, they're going to be little gaps in the backbone between those Okazaki fragments. And so to glue or to reattach the backbone of the DNA, that's where the ligase comes from. So you can think about ligase as glue or like a sewing machine or something that just comes and like repairs little gaps in the DNA as it's being made. Okay, so that's our ligase. All right, last thing to talk about in eukaryotic DNA is a telomere and something called telomerase. Okay, this is a lot of reading. Sorry about all the words, but basically what happens is, and this is only in eukaryotes, okay? This is only for those linear, if we have a long linear strand of DNA, what happens is every time it's copied, it doesn't go all the way to the end. So a little bit of the end is lost every time DNA is copied. In order to not like lose, so if you think about your skin cells or something that's like constantly regenerating, right? We're gonna have to copy DNA a lot. So we don't want the ends to erode over time, because if you erode too much, you're gonna start hitting important genes and destroying the DNA. So what eukaryotic cells have in their genome is a little segment on the end, which is called a telomere, okay? A telomere is basically just sort of nonsense. It's non-coding DNA. It doesn't do anything except create like an extra piece, extra DNA on the ends, so that when you do replicate and you lose some of the ends, you're not losing anything important. So it's basically like a little buffer zone. It's non-coding DNA, meaning it doesn't code for protein, and it protects the ends of the molecule from getting eroded. So what's interesting about telomeres, right? So I mentioned that every time DNA is copied, telomere is eroded and shortens. It's actually a signal for the cell to die. So this is really cool. If a cell recognizes that the telomere, this area here on the end, is getting really, really short, and that if you copy the DNA again, you might actually start to eat in at the actual information. Those really short telomeres tell the cell, hey, I think we hit the end of our lifespan, we need to die. And that programmed death is called apoptosis. Okay, apoptosis is a programmed cell death. It is a way for the cell to, basically kill itself. Is a bad way of saying it, but it will just like, instead of being a problem, instead of being this like mutated, instead of making a mutated cell with like missing chromosome, the cell will just go ahead and die through apoptosis. It's a very normal thing. And so that's where this enzyme comes in, telomerase. So when your cells are dividing, you lose a little bit of that telomere, this enzyme telomerase comes in and extends it. It just makes more telomere so that it extends the lifespan of the cell, right? It makes the cell able to divide more. And so you get more replications and extends the life. So that's the importance of telomerase enzyme. We have this enzyme to help elongate the telomere region so that the cell can have more replications. There is a downside of this, and the downside is that when you have telomere extension, right, if you're adding more telomere, the cancer is basically uncontrolled cell division. You get cells dividing and dividing and dividing. Their cells have abnormally high telomerase activity, meaning that the cancer cells keep putting on more and more telomere, and so the cell never gets the signal to apoptose or to die because the telomeres keep their nice big long length because cancer cells have sort of an abnormal telomerase activity. So something to think about. There's a lot of cancer research in this telomere area. So that's interesting, but only in eukaryotes, right? Because we have a linear strand of DNA. So this is just a way of showing you that if we have a parent strand, we have semi-conservative replication, just that we're losing some of that telomere with every, with the leading strand and the lagging strand. So just a picture of that. All right, what's different about a circle, right? So when we talk about prokaryotic DNA replication, it's called rolling circle replication. And basically you have an area where this double-stranded molecule is nicked, so it just breaks open slightly. And then your DNA polymerase comes in and it copies the DNA, right? So look at this blue one that's coming out. So it's a displacement of the nicked strand. So the nicked strand comes out like this. And I have a word here called single-stranded binding proteins. So because this is one strand of DNA, because it has those hydrogen bond, right? It has those loose nucleotides that wants to hydrogen bond. This is called sticky. You can kind of think about DNA as like two sides of Velcro, where like given the chance, the two sides want to stick together. So we want to keep this as a linear one strand. And so we don't want this to go back and like flop back, right? And to like make a double strand again. We want to keep it open. So these little proteins called single-stranded binding proteins will come and just attach to that single strand to keep it open. So there's no way that this one strand can go back to the other strand, right? And hydrogen bond again. So it keeps it open. So that's what our single-stranded binding proteins do. And you can see that the DNA polymerase is going to work its way around, right? And then as it's doing that, right, you're going to get this original, like the original circle with this red going. You're going to get the DNA polymerase going all the way around, making that other strand. And then the one that's nicked, right, it's going to also have another DNA polymerase add on to it. So it'll just also attach, go around the circle. And so the same thing happens where you have semi-conservative replication. It's a lot easier and simpler. There's no bubbles, there's no, forks, things like that. So if we look at the list here, we still have our DNA polymerase 3, okay, same as ours. We still have helicase to open up the double bonds of our DNA. And then we still have ligase, because when you're done with the copying, you still need to attach the backbone of the new circular piece of DNA, right? So there is a video here that I'm not sure if I can, let's see if I can make it work.
In this image, the DNA from a bacterium has been gently released from the cell. Most of the DNA is from the bacterial chromosome, but small circular DNA molecules called plasmids also exist. For many plasmids, replication is not tied to chromosomal replication. Many plasmids replicate autonomously by a method called rolling circle replication. In rolling circle replication, a replication initiator protein called Rep-A binds to a section of the double-stranded DNA called the origin of replication, or ORI. Rep-A is encoded by a plasmid gene. Rep-A nicks one strand of the DNA and holds on to the 5' end of the strand. The 3' end, with its free hydroxyl group, serves as a primer for a host DNA polymerase to begin to replicate the intact complementary strand. The Rep-A initiator protein recruits a helicase that unwinds the DNA. As the DNA unwinds, it becomes coated by single-strand DNA-binding proteins. As replication proceeds, the nicked strand, which continues to be covered with single-strand DNA-binding proteins, progressively peels off until replication of the intact strand is complete. The two ends of the nicked single strand are rejoined by the Rep-A protein and released. DNA ligase seals the nick in the double-stranded molecule. The single-stranded DNA can now be replicated. A region of the DNA becomes looped, allowing RNA polymerase access to the DNA to form a primer. Host DNA polymerases use the primer as a starting point for the synthesis of DNA. Finally, DNA ligase seals the remaining nick, resulting in a double-stranded plasmid. Each of these plasmids can undergo replication again by the same method.
Okay, so that was a nice, animation of rolling circle. So I hope you saw that the use of ligase, we talked about ligase before with our eukaryotic genome too. It's the same enzyme and same function to glue that backbone. Okay, so I have for you just a side-by-side comparison about DNA replication. So for DNA, prokaryotic cells are circular, eukaryotic multiple linear. Origins of replication, there's only one where the nick is formed. There's many, right? We have to make many bubbles. Replication bubbles, there's no bubbles in prokaryotic, this rolling circle. For a eukaryotic, right, remember we had many replication bubbles. Each bubble has forks on either end. We have helicase, which unwinds, right, and breaks the hydrogen bonds. Same thing for eukaryotic cells. DNA primase adds a small double strand to allow DNA polymerase to bind. Same thing here. And actually, this is wrong. I just really quickly, just because you don't have to know this, but it's actually RNA primase. All right, but let's just call it primase. Why not? Primase puts the primer on. Single-stranded binding proteins, I did not mention it in eukaryotic, but it's the same. So we have the same binding proteins to make sure that the double helix doesn't, once it's separated, it doesn't go back to a double helix. So this is the same. Ligase does the same thing in both of our DNA replication. For the leading and lagging strand, right, that doesn't happen with the rolling circle. It does happen when you have a linear DNA molecule. And then telomeres, right? We didn't talk about that. Prokaryotes don't have telomeres, but eukaryotic cells do, right? Because it's a circle, so you kind of like don't need to protect the ends. But in eukaryotic cells, since we have a linear DNA, we do need to protect the ends from being eroded. And so this is just an overview, a cute picture of our helicase, which unwinds the DNA double helix. Then we have our polymerase enzyme that synthesizes the DNA. I think that's why it looks like a little tool, a repair person. Primase puts the primer on, right, so that it creates a double strand so that DNA polymerase can attach. And then ligase is our little glue, right, that fixes those little backbones to make sure our DNA molecule isn't broken. And that's the end of part 1.